Our next two speakers will be introduced by their supervisor, Dr. Ryan McKellar. Ryan is the curator at the Royal Museum in Regina, and he'll provide context into their work and how it fits into the bigger picture at their museum.
Thank you for having both myself and the students as part of this talk series. It's fantastic. I guess in terms of larger scale stuff that we're working on or how this fits into the bigger picture, the work at the RSM is sort of done by a motley crew in Saskatchewan from many, many institutions. And the two folks that we're going to highlight today are Ben Egan and Katie Kreutzer. But a lot of the material that we're working on comes from things like dinosaur dig sites or coal deposits that are being worked on by much larger groups from places like the Phil Currie Museum in Northern Alberta, places like the University of Regina, University of Saskatchewan, and University of Alberta too. So it's a big collaborative effort. Katie's work centers on some of the amber from coal seams, but she's also working on amber from dinosaur bone beds. She is working on her honors undergrad thesis project on the topic that she's going to talk about today. So I'll let her talk about that without stealing any of her thunder. Ben just finished his master's degree at the University of Regina, working on comparing amber isotopes or stable isotopes of hydrogen and carbon to stable isotopes from dinosaur tooth enamel to trace things like food webs. So basically working out the types of plants or the offset that happens when herbivores eat plants and then when the dinosaurs eat each other, sort of thing, as you go up through the food web. And he's extended that for his PhD project, which he's starting up this winter. And overall, what we're trying to do is create sort of a large-scale picture of amber deposits through time. So just like Andre's excellent talk here, we're stretching from sort of the foremost formation all the way up to through the Paleocene, or the last part of the Cretaceous and up into the Paleocene. And what we're focused on is what is happening around the extinction event at the end of the Cretaceous period. The little yellow dots that you see on the screen here are occurrences for amber deposits through time. The red ones, the red arrows, are the sites that have been published on with inclusions in them, and the yellow arrows are pointing to sites with inclusions that we haven't published on yet. So you get some sense that there's a fair bit of density of amber sites through time, and it's not just about the insects. Some of the other work that we can do with this material looks at chemistry and things like that to work out ancient environments. So beyond looking at ecological connections, we can do things like tracing water through time or precipitation patterns through time. And as you go up through your stack of sedimentary rocks in Saskatchewan and Alberta, you can basically see conditions changing between conditions like you'd see, or rainfall conditions like you'd see in Dallas, Texas versus Edmonton. So the comings and goings of the Western Interior Seaway show up in the chemistry or geochemistry of some of these resin samples. And this is particularly neat and relies heavily on sampling from museum collections and also a lot of outside sources. So groups like the APS have been really crucial in some of these studies. So we've actually had the luck of working with APS folks at sites down by Rolling Hills. Max and Harold in the past have had us down for a short course and then taken us out to site by Rolling Hills. It was long, long ago though, when my kids were much smaller and much cuter, but it was a blast. And it's exciting for me to see the students working in the lab now working with your team as well or presenting to your team as well. So without much more ado, I will pass it off to Ben and he can take the lead here.
Today I'm going to walk you guys through our research project in describing a new species of Staphylinid beetle from Dominican amber. I've got it here on the slide, and as you can see, we were quite fortunate in the fact that we had two beetles of the same species preserved in the same amber piece. Really helped when it came to describing these beetles. Now, these two beetles are very important to me personally and academically, as they are a big reason I'm even in Canada in the first place. Unfortunately, you're going to have to hear a little about me to understand why. So hi, I'm Ben. I grew up in England, and it's where I did my bachelor's in geology at Durham University. My main background, of course, mainly being in earth sciences. Whilst at Durham, I did a 3-month Mitacs Global Link internship at the Royal Saskatchewan Museum. Some of you might be familiar with Mitacs. The Global Link project is essentially designed to get international undergrads into Canadian institutions to do a research project and give them a taste of what graduate studies is like. The initial project was amber inclusions across the KPG boundary to essentially look at insect diversity, although we pivoted a little bit for the project to describe this new species. Something a bit outside my comfort zone, not really being a biologist or an entomologist. That experience was a big reason why I came back to my master's, not necessarily in entomology, but as Moran mentioned, in amber geochemistry and looking at dinosaur diets. All right, hopefully that's enough about me. What is it like to describe an insect in amber? Or for some of you, it might conjure up imagery a la the first scenes of Jurassic Park. where they are in that Dominican amber mine, and they all gather around this beautiful big piece of amber to look at the mosquito inside. Of course, in the movie, they are more interested in finding that dino DNA, which of course we now know doesn't preserve nearly as well as they thought in the movies. But in real life, they would be just as excited to see that insect inclusion. Of course, a lot of this is dramatized for Hollywood. One of the sad things I was just recently reading up on is that most Dominican amber mines you can't stand up in. It's quite often that they get waterlogged as well and cave in. So it's quite a dangerous profession to do in the Dominican Republic. Unfortunately, I never got to experience this part of collecting our amber sample. By the time I got it, I'd already exchanged multiple different hands. The most discovery we did was essentially looking on the shopping page. The amber market is huge, whether that be in the form of jewelry or just buying inclusions straight up. One of, quite often actually as well, you'll find posts advertising potential new species to be bought at a lot cheaper prices than you would think. Of course, this comes up with the caveat of who do you trust, and if it's a trusted seller that will sell you fake amber. When it comes to trying to spot fake amber in a lab, we generally have a motto. If it's too good to be true, it probably is. A lot of amber fakes are perfectly placed and in perfect condition, very unlike what we see in actual amber fossils. Another way to spot some of the cheaper fakes is by looking at the resin mold lines. Often you'll see where they've tried to graft either pieces of amber together or even just create a mold for the resin using actual modern day insects or even rubber molds. Another really cool way I found, I've learned during my master's to identify fake amber is by shining a UV light on them. The chemical composition of amber essentially is composed of aromatics, which react to that UV light and re-emit a blue or green glow. How can we be at the stage where it's so easy to essentially find a new species that you can just buy it online? Well, it's down to just simply how many species of insects we have in the modern world and throughout the past. As of 2022, we've described over 1 million insect species. This makes them not only the largest species group of animals, but insects have more species than certain plant and fungus groups. We also still think we've got a long way in describing all of insects. Current estimates place the potential amount of insects alive in the world today at anywhere between 3 million to 30 million, with most of them thought to be in the tropics. In fact, the biggest hurdle to describing new insect species isn't our approach or the fact that we're not finding these species, it's the fact that we just don't have enough people working on it. Now, talking about the amber piece we looked at. So it comes, of course, from the Dominican Republic. And for those who don't know, that's on the island of Hispaniola in the Caribbean. Now, most amber deposits on the island are Miocene age, anywhere between 20 to 15 million years old, although some older age estimates exist based off coccoliths. Now, what's great about Dominican amber is it's often quite beautiful and clear. It's very easy to look into and describe insects from. Of over 400 currently being described. Some cool things about the assemblage, like a lot of island ecosystems, you find a lot of species uniquely only from Hispaniola and the Dominican Republic. Also very interesting to see when you compare some of the insects you find in Dominican amber, often you can find a modern counterpart. They're essentially the descendants have not undergone much evolutionary change. Externally, they look quite similar to their ancestors. So why did we pick this particular amber piece to look at? We had plenty to choose from those online. But what we found really interesting about this amber piece, and I tried to circle it here in this picture, is that the adadagus, or the male genitalia, had been exerted outside its body. Normally in life, the genitalia remains inside the body until the animal is ready to mate. Now, this can happen in amber in a number of ways. Often when animals are engulfed in amber, their insides get squished and become outsides. We didn't initially think this was the case for this sample. The fact that we had two members of the same species within the same amber piece made us think maybe these beetles were trying to mate. This is one of my most favorite things about amber, its ability to capture a moment in time and give us an idea of some behavioral patterns of these animals. I've got a few examples here on the slide. So in the left-hand picture, you see an example of a parasitic nematode erupting from an ant. Often you'll see YouTube videos of them doing it in praying mantis. It was a really cool example of this parasitic relationship in Baltic amber being around for over 40 million years ago. In the middle picture here, we've got a very kind of morbid specimen. These are lacewing larvae. They have actually just hatched from their eggs and become immediately engulfed in amber, dying very quickly in their life. And finally, in the most right-hand corner, we have an example of a spider trying to consume a wasp in amber. There are numerous other examples, and it's one of my favorite things about amber, that ability to show those ecosystem dynamics quite spectacularly. Another great thing about amber is that it's able to preserve your specimens three-dimensionally. We take advantage of this at the Royal Saskatchewan Museum with our partnership with the Canadian Light Source in Saskatoon using their synchrotron. Even before I came to Canada, one of the students on the paper, Alyssa Lowen, went through and CT scanned a lot of our amber inclusions, essentially taking X-ray slices through the entire amber piece. What we are then able to do is create 3D models, which greatly helped in describing our beetles. We're able to manipulate these models and look at them from different angles that we wouldn't be able to if we just had the amber piece. We're also able to zoom in on certain parts of the animal that are allow us to more accurately identify it. And one of the best things of all, you're able to look at internally in the animal. Now, sometimes this can be a bit iffy. If amber has infiltrated the inside of the animal, the X-ray won't really pick up anything. But we were quite fortunate in this case that a lot of the internal anatomy of these animals was still preserved. The unfortunate reality of this, though, was that when we were looking at what we presumed to be our female beetle, we found that inside the beetle was male genitalia. It was actually a male, which kind of shut down our hopes of advertising these beetles as a mating pair, which was kind of sad for me. But another great benefit of creating 3D models is that you can 3D print them. At the Royal Saskatchewan Museum, we have two main types of 3D printers. A filament one, which you're probably more familiar with, and most hobbyists have, where essentially you take molten wire plastic and go around to build up layer by layer. And we also have a resin 3D printer, which I've put on a time lapse here. Essentially, what this does is lowers a plate into a vat of resin and flashes a light. The resin actually reacts to that light and hardens to the plate and slowly builds up the model over time. These are great for helping us describe the beetle, but also great for programming. Although I did learn a very tough lesson when I gave it to a class of 7-year-olds, and my beetle came back a lot worse for wear, missing a few of its legs. For all that, we were able to narrow down our beetles to Staphylinid, also commonly known as rove beetles. I say narrow down. Staphylinid is the largest group of beetles, which beetles themselves is the largest group of insects. We had to deal with potentially over 66,000 other species. For such a large group, they are very varied as well, ranging in size from anywhere between less than a millimeter to 35 millimeters, and display various different ecologies and behaviors. One of the biggest struggles we had in trying to identify our beetle was the fact that it didn't really look like a traditional rove beetle or what we'd expect it to be. A few of the really cool examples of Staphylinid on the slide here. In the top left-hand corner, we have the devil's cultureless beetle, known in the UK for bringing a sign of devilry and witchcraft. And that's down to just how mean this beetle can look. It's got a very unique body plan and a very interesting defensive posture where it raises its abdomen, as you can see in the picture, almost akin to like a scorpion. We also have the hairy rove beetle on the top right-hand corner there. This beetle is known for its unique ecology of laying its eggs in carrion, not only to feed on the meat of the rotten corpse, but to potentially let its larvae feed on any maggots from flies in that corpse as well. Another cool example, we have bee mimics. These Staphylinid hang around near hives. And we also have ant mimics, ant-like stone beetles, which our beetle belong to. The problem I have with ant-like stone beetles is a lot of them look very similar to each other externally. Here I've got 3 different beetles, all from different genuses. They look very much the same externally. Fortunately, though, we did have one advantage. The fact that one of the best ways to pin down which group these beetles belong to is by their genitalia. So again, that male genitalia being very useful in identifying which group we belong to for our beetle. It's to the point actually where it can be quite difficult to identify them externally, that often female members of these groups are quite difficult to place properly. So after staring at beetle genitalia for much longer than I'd like to admit, we're able to place our group into Alloraphes. Now, Alloraphes is a small group of beetles, only about 14 members, and then 15 with our new species. And they're also small in stature as well, being generally less than a millimeter, same as our beetle. These beetles are only really known from Central and South America, although in the past people had speculated that they found some in Africa. That turned out to be another genus in the ant-like stone beetles grouping. The ecology of these beetles is generally that they like to live in leaf litter and they consume mites, they're predators, which kind of begs the question, how did they end up in our amber piece? Obviously, amber coming from trees, it's a bit separated away from the forest floor. Well, looking at our amber piece, it was fairly clear, so it's not like the resin fell down and engulfed them like so in the leaf litter. It's much more likely the fact that the small size of these beetles basically meant that they got carried away into the wind and blown into our resin and trapped. Some of you may have noticed I've not really said the name of these beetles. I've kept going, these beetles and our beetles. And that's because it took me a very long time to try and come up with a name for these guys. I potentially suggested naming them after a family member or even naming them after where they were found. Instead, we decided to do the kind of the better thing and name it after its morphology. So unfortunately, we're looking at the genitalia again. And one of the things that stood out to me most about the genitalia was these little paddles on top. I thought I had very fond memories of growing up and using a tuning fork in primary school in music class. And I thought the parameres, or these little paddles, represented that tuning fork very well. So we went with the name song fork or cantus furca. So the full name is Alloraphes cantus furca. As a little aside, I was quite insecure about this name, so I had to go around and show it to all my friends, and they got very sick of seeing beetle genitalia constantly, just as I have. And they gave me a very mean nickname that I've been advised not to repeat here. So what is the point? What have we got from this study? With this study, we have the first Alloraphes beetle from Hispaniola. It is also the first fossil Alloraphes extending this entire group to the Miocene. What's also, I find, really interesting, given the fact that a lot of Dominican animals or insects don't seem to change that much from their Miocene counterparts, and the fact that these beetles are small and like to live in leaf litter, there is a potential that their descendants are alive and well on the island today. Just need some more people to look at it. Thank you very much for your time. And does anyone have any questions?
I was just wondering what kind of chemical characterization you do on the amber, and if any of that information, like say XRD, could give you any useful hints, just in any sort of determination of species and things like that.
Oh, for the beetle, or for what the producing tree?
For the beetle, if there's anything, because you said it's mainly like polyaromatic hydrocarbons, things like that, right?
Yeah. Well, the amber itself, I wouldn't know. I've not really looked at the chemistry of insects. I know a bit about the chemistry of amber in terms of identifying that, but I won't go too much in depth for that because I believe that's part of Katie's talk that she's going to get into. But yeah, no, I've not really looked into that. No, it's mostly just been looking at keys and getting some advice where help needed.
Okay, cool. Thank you.
A couple of questions. How come Baltic amber, there's such a market for it? I mean, it's sold everywhere. Is the Dominican amber retailed, or is it just scarce? Or, and then the other question was, when winds blow up from Africa and go all the way across to the eastern coast of the United States and then the Caribbean, are there any African beetles that are found in amber, or just found there in modern time? Or is it just too much of a difference, too much wind? And
I'll go first question first. So, from my understanding, Baltic amber is just one of the biggest amber markets. What's also really cool about Baltic amber, it gets spread across the entire Baltic region, coming from multiple different deposits, while Dominican amber is kind of just confined to the Dominican Republic. So that would be my guess of why Baltic amber is such a much more prevalent deposit. With the idea of all these potentially insects getting blown across, I think it'd be very, very rare for them to make it all the way to continental US. I imagine a lot of them would either die along the way. So I don't know how likely it would be for them to even preserve an amber. You're probably talking a very, very rare chance. So I feel like it's more likely that they'll be expatriated from either so for instance, the Dominican Republic, more likely that'll be coming from Central America, and that's how they arrived there, rather than coming all the way from Africa. But that's probably how some of these beetle groups first came to the island in the first place.
Okay, thank you.
Oh, thank you.
Thank you, Ben. I think that's all the questions we have here. So thanks again, Ben.
Perfect. Thank you.
And Katie, if you're ready, please go ahead whenever you want.
This is my bachelor's thesis. It's working on Paleocene amber from Estevan, Saskatchewan. I'll be talking about the geochemistry and also the bioinclusions that we found in this site. So first off, Ryan already introduced me, but I'll just give a brief little synopsis here. I'm Katie Kreutzer, and I'm in my last year of my undergrad at geology at the U of R. And I've been a student researcher at the department for the last few years, but it was only just this last summer that I started working with the Royal Sass Museum and with Ryan McKellar, especially on my undergrad thesis with amber. My area that I'm really interested in is combining geochemistry and paleontology, and especially using stable isotopes on amber. When I talk about amber, what do you typically think of? Most of you will probably conjure up an image of that Jurassic Park scene where the scientist drills into the amber to extract the DNA. Some of you might think about amber jewelry because it's found all around the world and very extensive. And some of you might even think about amber ale. So all of these are different human interpretations of amber that we see. But what really is amber? Amber is a resin. It's not a sap. That's the main difference, is that a sap is like the blood of the tree. It carries the nutrients of the tree, where a resin is more of like the scab of the tree or the pus of the tree. It's the protective barrier that seals the wounds. What's important about that is that resin is only produced when the plant is stressed in some kind of way. And when the resin is fossilized, then it becomes amber. So one of the key differences between what Andrea was talking about with impression fossils versus amber is that impression fossils are 2D, where amber is 3-dimensional. And as you saw in Ben's talk, we can actually look in all dimensions and CT scan and whatnot. Another really great thing about amber is that its chemical structure is actually really stable, which means that it's not likely to be altered through diagenesis. And this makes it a great paleoenvironmental proxy. So that's where the stable isotopes comes in. One of the really difficult things with specifically the inclusions in amber is that there is a large gap, an amber gap, from about 70 million to 55 million years ago, where we actually have very little amber preserved across the world. And we don't know exactly why we just don't have any amber, but it really limits our understanding of arthropod evolution across the KPG mass extinction event, because amber is one of the main ways that we preserve arthropods. So we don't understand how different groups succeeded in the extinction event, how they bounced back, which groups survived and when, and the timelines. It's all very unclear because of this amber gap. So that's one of the goals of the RSM, which Ryan had mentioned, was filling in that amber gap. For my work, we do have to talk a little bit about chemistry. Every single element has a slightly different version, which has slightly different weights. So for example, carbon-13 versus carbon-12. Carbon-13 has one extra neutron than carbon-12, which means that it's actually physically heavier than carbon-12. Now, life is very lazy. Biology will take in more of that lighter version than the heavier. And this process of taking more of the lighter than the heavier is called fractionation. So when we apply that to the fossil record, we see that a plant is only really going to take in a lot of that heavier carbon-13 because it has to, because it's stressed in some kind of way. So when we look at the ratio between the two, we can see if a plant is stressed or if it's healthy. And when we apply that to hydrogen, that fractionation has to do with precipitation and rainout cycles. So when a cloud is moving across the continent, the heavier hydrogen is going to fall out first because it's physically heavier. So as that cloud moves across the continent, it's becoming lighter, and the rain is becoming lighter that's raining out. So we can look again at this ratio, and we can understand how coastal or how distal we are from the ocean. And this has big implications for the Western Interior Seaway in the Paleocene. Okay, so where is my study located? So we looked in the southeast corner of Saskatchewan, near the city of Estevan. This area is in the Ravenscraig Formation, which is approximately 63 million years old. We have palynology estimates for this, but we don't have any radiometric dating at the moment. So that's something we're working on. But it's essentially equivalent to the Pascapoo Formation in Alberta. This formation, the Ravenscraig, has been typically described as a meandering river system with very extensive swampy backwaters, which created all of the coal that is being mined in southern Saskatchewan. And as a byproduct, some of the amber that is preserved there as well. So our study site is a road cut just outside of the city, and it's part of the Estevan coal zone, which is the brown area on the map. Each coal zone has individual seams. So you have the formation, and then you have the individual coal zones. And then within that, you have specific coal seams. So the Estevan coal zone has 4 coal seams from A to D, A being the lowermost and D being the uppermost. And the D seam is where we found almost all of the amber. What did we end up doing? Well, we went out to Estevan this summer, and we collected a bunch of amber with just taking little tweezers and putting them into vials and just filling up as many vials as we could. And then when we got back to the lab, we started working on a few different things. So stable isotopes, FTIR, and then inclusions. I'll just briefly explain each of these. For the stable isotopes, we selected clear pieces of amber to make sure there wasn't any kind of alteration with them. And then we sent them off for mass spectrometry. The carbon was sent off to we did it in-house at the U of R, and then the hydrogen was in Ottawa at the Jan Visor lab. So essentially the mass spec vaporizes the solid amber and turns it into a gas that we can analyze. The FTIR is actually, it's really neat. It's kind of like fingerprinting. It's molecular fingerprinting where you can actually take the machine scans infrared light into our sample, and then it detects which wavelengths are reflected and how strongly they're reflected. This reflection at different wavelengths is dependent on the underlying chemical structure of the material. So it tells us what the material is chemically made out of, which is really, really helpful for understanding what the source of our amber is. What kind of family of plants are we dealing with here? And then lastly, the inclusions. We did thousands of amber samples that we embedded in epoxy and then screened. Very arduous process of screening each one and looking for any kind of biological inclusions in them. What did we find? So this is the carbon graph that I made here. And essentially on the left is the time scale that we're looking at, and along the bottom is the carbon values. And generally we have, as you go more negative, we have more or healthier, it's less stress. And then as we go more positive, it's rich, so it's more stress. Generally, that's how it looks. So what we're seeing here is that we compared our site to other sites around Saskatchewan and Alberta that the RSM has been collecting data from to fill in this gap. But we're filling in a gap within a larger gap, which is really neat. What we're seeing is that we're actually getting pretty similar results to some of those other sites, which, it's good. It means that we're not crazy out of the range of possibilities. We're pretty good that way. We're getting an average of about -23 per mil, which is actually significantly more stressed than modern plants. But I'll get into that in a minute. We don't know 100% why that's happening. But for our hydrogen values, same kind of graph here. We have the time scale on the left. And then on the bottom, we have, as you go more negative, it's less coastal, you're getting more inland. And then as you're going to the right, you have more coastal, you're closer to the ocean. So we kind of have to be careful when we're comparing, again, those same sites from Saskatchewan and Alberta, because they obviously have different positionings and they're going to be closer or farther away from the Western Interior Seaway. But I wanted to include them here anyway, just to see like, okay, we're within the same kind of realm of possibilities. We're not getting crazy outliers. But essentially what we're seeing from these seams in Estevan is that we're getting a significant to moderate coastal influence, at least more coastal than current day Estevan. So modern day Estevan has about a -100 deuterium value, where we're getting between -60 to -80. So that's between like Winnipeg and the border of New Brunswick and Quebec. So it's definitely some kind of coastal influence, at least significantly more than what we have today. This is the FTIR. I won't go in too much depth about this, but essentially it's like tea leaf reading where you're matching different peaks and trying to get them all to line up. What this is suggesting is that we're comparing the Estevan seam to modern Cuprocacea plants, and then also the Drumheller Valley amber, which we also know as Cuprocacea. And we have the diagnostic peaks on that graph there, and it essentially is just saying that, yeah, all of the peaks are matching up really nicely. So it suggests that the source is Cuprocacea trees, redwood. One of the most exciting things about this project has been the inclusions. The first inclusion and the biggest one so far that we've found has been a full adult mite. He's been lovingly dubbed Mite Wazowski. We haven't given him a name other than that. That is his official name as of right now. But thanks to some of the mite experts at the U of A, we've actually been able to identify the family to Stigmaidae, which has been really interesting for me to look into, because this represents the first arthropod found in Paleocene Saskatchewan. And because of that bioinclusion gap that we see, it's one of the very few arthropods that we found in the Paleocene in general. So that's been really exciting. One thing that I kind of recently looked to was trying to find the oldest Stigmaidae family of mite in the fossil record. And as of right now, the oldest I've been able to find is from this site in Ukraine that is between 35 and 38 million years old. And if you're playing the numbers game here, our site is at least 63 million years old. So if I've done everything right here and we've got the identification right, this is the oldest Stigmaidae family of mites that we found by at least 25, 30 million years, which is really exciting. So I'm going to keep looking into that. I don't want to say that it's like the oldest, but it looks promising at least. So recap. What does it all mean? So essentially, the carbon isotopes are showing that there is some kind of stress, that the Estevan forests at this time have some stress, and when we compare them to modern Cuprocacea plants. And this could be likely representing differences in environmental conditions. We do have higher temperatures in the Paleocene than modern, so that could be part of the reason. It could also be like plant stress due to insect attacks or wildfires or other things like that, or even water availability or water salinity due to the Western Interior Seaway. We're not 100% sure, but it could be any or all of those factors attributing to that. The hydrogen isotopes definitely show a moderate to significantly more coastal influence at the Estevan site compared to modern day. However, we do have to be careful just because differences in paleolatitude could also account for some of this difference. So we haven't been able to really outline where the bounds of the Western Interior Seaway are at this time. So that's something that I'm going to continue to work on and really refine down a little bit more. The FTIR, of course, was able to identify the family, Cuprocacea. And then lastly, the inclusions that we found represent the first arthropods found in Paleocene Saskatchewan, and with more samples on the way. I've got a few in the lab that I'm working on right now. I've got an aphid that I'm just finishing up, and a few other maybe mites, maybe bugs, maybe, I don't know, a few kind of mystery inclusions that I'll be working on to include in my thesis. So that is pretty much it for me. If you have any questions, let me know. I have to give thanks, of course, to NSERC, the Royal Sask Museum, and the University of Regina for funding for this project. And then I also have to thank my supervisors, Drs. McKellar, Velez, and Robbins, who have been super supportive of this project. And of course, the team at the Royal Sask Museum, Ben Egan, Caitlin Lindblad, Lauren Lindsay, and Aurora McLeod, who helped so much in this project. We had a few volunteers from the U of S who helped pick amber with us this summer, so gotta thank them. And then, of course, Louise Schneier at SAS Power, she was able to help us get land access to these sites. And she joined us in the field for a few days, and it was really great having her. So that's pretty much it for me. If you have any questions, please let me know.
Thanks, Katie. Are there any questions?
Okay, Jack, you got a question. 1 second. What additional lines of evidence, either sedimentological or paleontology, would you accept to see and assist in the interpretation for modern marine influence? That's a good question. Okay. So the difficult thing is that we are seeing from the sedimentology basis, it does look very terrestrial. And it's always been interpreted, the formation as a whole has been interpreted as terrestrial. So I don't know exactly what we would be looking for, because in the coals themselves, I've looked at some of the mineralogy and stuff. There doesn't seem to be a marine influence itself. So we don't think that it's like Estevan is right on the border, but there is at least some proximity. I still think that it's a terrestrial environment, but it's a question of how close is the Western Interior Seaway, right? So I don't think it's marine, but I do think that it's closer than what we previously thought in distance there.
I've got a question.
Yeah.
Really interesting talk. For
Thank
sure.
you.
Two questions. One, have you been able to compare some of these results with some of the amber that you find out by Grassy Lake?
If I'm not mistaken, I think that Grassy Lakes is like 75 million years. Ryan might know better. Let me.
About 78 or 80 at the top of the foremost formation.
Yeah, so it's between like 18 to 20 million years older. So with getting all of these different amber sites from across the timelines, it's helpful to see in that way the changes with the Western Interior Seaway is really interesting. And that's something that I want to work on a little bit more, is trying to see like redefining where the bounds of the seaway was. So I'm not sure exactly, like, are you asking about comparing the like isotope values or the inclusions, or which part are you asking about?
General similarities.
General similarities?
I think you've answered my question.
Okay, okay.
Going back to your presentation, that gap that you're talking about, that 16 million year old gap,
Yeah.
that's pretty fascinating. I'm just really surprised because, you know, with at the KT boundary, of course, 85% of life died out, and then gradually it resumed.
Mhm.
A lot of things were permanently extinct, you know, dinosaurs and ammonites, for example. But to have that long a gap across that extinction boundary is kind of mystifying.
Yeah, it really is. And it's a global thing as well. It's not just like it's a North American thing or like a Central American thing. It's globally, we have very, very few amber deposits from that time. So obviously very, very limiting to understanding what's happening, not only with biosphere, but also with the environment. And it just limits our understanding generally on everything.
Right. Okay, thank you.
Yeah, of course, no worries. I think Brandon had a question here. You had mentioned that you were looking at mass spec data and FTIR to identify the family of tree. What chemical compound were you looking at? So we weren't looking at well, I'll just say that amber is made of terpenoids. So that's kind of what we were trying to match that way if you're looking for a chemical compound. But the FTIR gives us like a range of wavelengths, and we're matching different wavelengths in that way from the infrared light being reflected off of the sample. So that's the FTIR part of it. And then for the mass spec, that was for the isotopes. So it's how do I explain it? It's taking the solid amber and vaporizing it into a gas, and then we're looking at either the carbon gas or the hydrogen gas and seeing how much we have of the carbon 12 versus the carbon 13. And that's how we're getting our delta 13 values or our deuterium values. So I hope that clears that up.
I think it does. I don't see any other questions here, so thank you, Katie.
Awesome. Thank you so much for having me.
Yeah, and so thanks, Andre, Ryan, Ben, and Katie. Thanks Thanks again.